Hi everyone, and welcome to this video on mitosis and meiosis for KEEP Unit 1. So 90% of the information that I will be speaking about in this video, you already know from Form 5 when you did mitosis and meiosis. There's just a couple more steps that we will go in depth in. So it should be a very relaxing video for you and just a revision of what you already know. Okay, so we know that mitosis is a type of nuclear division which occurs during all growth. So when cells are going to divide, chemicals are secreted during the interphase, which is the first part of mitosis essentially, and they either stimulate or inhibit cell division, and they also regulate the rate at which the division would occur. Okay, so now after this, we know that the chromosome will be replicated. There'll be two identical molecules, and we go from the chromosome to the chromatin, then we go to the chromatid, and so on and so forth, right? We know about it, and I'll show you all the steps as well in this video, okay? So skipping the introduction, let's go straight into the interphase, which is honestly the only new thing that you really need to know. So during interphase, there are three sub-steps, essentially. So G1, S1, and G2. There's no S2, so you could probably just say S instead. And the entire interphase lasts about 24 hours. So G1 begins right after the previous mitosis. It's like a cycle, right? So chemicals are synthesized, and they would determine whether or not the cell will divide. So the cell has very high energy levels at this time, and ribosomes are synthesized to replace components that were lost in the previous mitosis. Okay, so in G1, the chemicals are synthesized there. In S1, so you could see G1 here, right? In S1 now, which is this portion, a replica of each chromosome is made. So you get the identical copy of the chromosome, and then it's called chromatids from chromatins, right? And they are joined at the centromere. We all know this, I'll show you diagrams after. And then finally G2, which is, as you can see, is a short process. There's a high amount of energy in G2, and it produces more mitochondria to keep the high energy going, and chloroplasts as well, if this was a plant cell. And spindle fibers begin to form here. So we know about the spindle fibers, and they are very important in pulling apart the chromosomes during division. Okay. So this process of mitosis essentially follows G2. Okay, and we know these steps, and we have our little thing here, IP mat. Okay, so that was interphase, which is technically not mitosis yet. So mitosis essentially begins at prophase. So here we have the summary. Okay, so during prophase, we have the shortening of the chromosomes. The nuclear membrane will disappear around the nucleus of the cell, and centrioles move to opposite poles of the dividing cell. So if you look at this small diagram here, you could see in prophase, you could see the centrioles moving to the opposite poles, right? So homologous chromosomes do not associate with each other, and this ensures that there is no opportunity for exchange, unlike in meiosis, as we will come to very soon. In metaphase, the chromosomes are positioned around the equator of the cell, and they are held in place by the attachment of their centromeres to the aforementioned spindle fibers that we are talking about. So here we have metaphase, and you can see that they are at the equator of the cell, and they are held in place by the spindle fibers at their centromeres. In anaphase, one copy of each chromosome will end up in each daughter nucleus. They are pulled apart, right? So the spindle fibers shorten and pull the chromatids apart. And you can see that here, you can see that they are being pulled apart. As I said, it's the same thing that you would have done in Form 5, right? And in telophase, the nuclear membrane will reform around each of the daughter cells, and they become closely packed together. The cell membrane constricts into two, it divides the cytoplasm, and it's called cytokinesis. That's a new word there for you, right? The process of the splitting is called cytokinesis. All right, and that's just a summary of something you already know, which was mitosis. So meiosis, more of the same, okay? So meiosis is a process of nuclear division in which the chromosome number is halved, right? So in mitosis, the chromosome number remains the same. In meiosis, it's important, the really only function of that is for the formation of gametes. And there are two major events that occur during meiosis. So one is the separation of homologous chromosomes. That is important because they need to exchange information. Each daughter cell will be different, right? And this occurs in meiosis I. And finally, there's separation of chromatids during meiosis II, right? So in meiosis I, we ensure that variation occurs, right? So these are some terms that you should be aware of in meiosis that would have not been in mitosis, okay? Right, so meiosis I. So the homologous chromosomes would lie side by side, and we call it synapsis. Each pair of chromosomes is called a bivalent, right? So pairing off enables contact to be made at certain point, right? And between non-sister chromatids, and that results in a crossing over. So it looks like you have one chromatid here, all right, short and long, and then you have a next one very close, and there's a crossing over right there, and that is exchanging of information, right? So honestly, the steps over here are essentially the same as mitosis, except that there is some crossing over occurring, all right? So we don't need to go through this all over again. In fact, you could just screenshot and read it yourself without me having to dictate it for you. But here is a very nice diagrammatic representation of meiosis, and it's a good summary. I like to use it, and you could practice to draw it as well as take a look at the steps in meiosis I and meiosis II on the right hand side. All right, the final thing that we want to talk about are chromosome mutations. So chromosome mutations may involve structural changes in a chromosome, or it could involve changes in the number of chromosomes, so either structure or number. So when structural changes occur, it can involve deletion of certain genes, duplication of genes, or something like we like to call translocation, or transfer of one part of the gene to another, okay? So translocation is an important one. So sometimes an entire chromosome may become attached to another one, right? So this is seen in Down syndrome, where there are 3 copies of chromosome 21, okay? So chromosomal mutations can also involve changes in a number, so it's called aneuploidy. So in Down syndrome, we call it triploidy because there are 3, okay? It may also involve a change in the number of sets of chromosomes. So an organism can have multiples of the haploid number of chromosomes, okay? Right? So this is your last part of meiosis and mitosis here. I hope you guys enjoyed a short video. It's just a recap.